everyone. I'm Victoria Popic, and I am a PhD student in Serafim Batsoglu's lab. Today I'm going to briefly talk about our work on cancer phylogeny inference using multi-sample somatic variants. So as we know, cancer is driven by the accumulation of somatic mutations, and if we look inside each tumor, we'll find that it actually consists of a mixture of cell populations, each one carrying a distinct signature of the accumulated variants that confer some kind of fitness advantage to the cells. In order to characterize this heterogeneity better and allow us to infer a more accurate cell lineage of the cells inside the tumor, many recent studies have now extracted actually multiple samples from a particular tumor, be it from different regions of the tumor or from different points in time in the tumor progression. So one of the projects that actually looked at multiple biopsies of breast cancer data at different progressions and in the breast cancer evolution was the work of our lab with, in collaboration with the School of Medicine at Stanford, in particular Professor Rob West and Aaron Sido. And in this particular project, they were looking at reconstructing the lineage trees and defining the heterogeneity of breast cancer samples. However, they were doing that manually. So we decided to further this work and provide actually a computational technique that would allow us to scale to much more samples, many more mutations, and to handle this type of problem much faster. Anyway, so this is a project called LICHEE, again in collaboration with Aaron Sido and Rob West, and particularly with postdoc Raheli Salari in our lab. And the goal of this project was to have a fast computational method that would allow us to infer cancer lineage trees and decompose each cancer sample into its subclones, while using multi-sample variants such as somatic SNVs. Okay, so the input data to our method is essentially a matrix where we have each row corresponding to a different single nucleotide variant SNV, and each column is actually the variant allele frequency of this variant across the different samples that we have access to. So again, this could be different regions of the same tumor, for instance. And our goal is then to take this data and to infer kind of a hierarchy or ordering of these mutations, so place them on a lineage tree, as well as decompose each sample into the constituent subclones that we infer, providing the proportions of these subclones in the sample. And just a small note here is this method can be generalized to work with other variants. However, that's assuming that we have access to something that would estimate the fraction of the variant in the samples, which is actually a very hard problem. So for things like SVs and other type of variants that are more complicated, this is hard to estimate, but there's a lot of work on that. Yeah.
How do you generate this matrix?
So this is directly following from the sequencing data. So we just compute the number of reads that have the variant as over the total number of reads that we have for covering a particular mutation, right? So this gives us just the raw frequencies right now. Yeah.
Small values like 0.1. There is a possibility that there are
it's error. Yeah. Yeah, definitely. So we have our own heuristical method that actually will look at the different variants and the frequencies and try to estimate whether this actually is a true variant or not. I'll talk about this in a second. But this is actually a challenging problem. So we do work with raw variant allele frequencies the way they are, but there are actually a lot of dedicated methods that have specifically been designed to call variants given very low fractions of reads that cover it. So it's such a challenging problem. So
genome data or exome data?
So we are looking at deep sequencing data. So that's why which allows us to have much more reliable frequency information.
It's most of the time it's just targeted sequence.
So right. So the data says that we analyzed actually. That I'll describe actually in a second. They validate a particular, like let's say a few hundred mutations with a very high depth, and I'll talk about this. But yeah, that's a very good point. This is a big challenge. So in our method, we make this kind of the big assumption that we make is that our data follows the perfect phylogeny model, also known as the infinite sites assumption, which basically states that the mutations do not recur independently in different cells. That means that if cells share a particular mutation, they must have inherited from a common ancestor. This is our main assumption. So this assumption allows us to formulate 3 constraints that directly follow, which you might have seen in literature. In particular, let's say we have 2 mutations, M1 and M2, and a set of samples in which they occur. And let's say we do know that they are actually there. Now, if a mutation such as M2 actually occurs in more samples than M1, it couldn't have been a successor of M1. Okay, this would violate the assumption that mutations don't recur independently in this additional sample as 2 here. So, and another constraint also directly following is that a mutation with a lower frequency value couldn't have really been a predecessor of a mutation with a higher frequency value. And here, assuming that we're not looking at a CNV region, and again, that we have reliable VAFs. Okay, and finally, the third is if we're looking at a group of mutations, one of the, let's say mutation M1 here, can only be a predecessor to a set of disjoint mutations present in disjoint subclones if their allele frequency is lower than the frequency, only if it's lower than the frequency of the parent. Okay, so in this case, M1 couldn't have been a parent to both M2 and M3 because the sum of these children mutations exceeds the frequency of the parent. And again, this is following directly from the perfect phylogeny model. Okay. So given these constraints, our goal is to find the lineage tree. So for the given set of mutations that we're looking at, find a lineage tree that satisfy these constraints. This is the goal of our model. And our main formulation is the following. So let's say that each mutation or a group of mutations that occur in the same subset of samples with the same frequency is a node in a graph. So we define all the nodes to represent mutations of groups of mutations with the same VAFs and presence pattern across samples. And what we do is we look at each mutation, each node in a pairwise fashion, and we place an edge in a network between 2 nodes if it's possible that the parent node can actually be a valid predecessor of the child node. So it satisfies constraints 1 and 2 that I've defined. So the parent is present in a superset of the samples of the child, and its frequency in each sample is higher than the frequency of the child mutation across the samples in which the child is present. So we look at all these groups, each one of mutations, each one will be a node, and we place an edge if there is a potential precedence relationship. So what we end up with is something we call evolutionary constraint network, which is essentially a DAG. And it encodes just all the possible precedence relationships, but in reality, it actually allows us to stream the search space for the true lineage tree substantially and have our method be very fast.
Victoria,
Yeah.
this can't be a real example, right?
No, this is just, I have a real example in a second. This is just...
Much more dense.
It can be. Yeah, actually, the example I have is close to this. It doesn't have to be dense, depending on the data, yeah.
Just a quick question. Some of your constraints come from set inclusion, exclusion, and there you might even get cycles, right? There you might even get cycles.
So we cannot get cycles. So basically, the parent node has to be a superset with a higher frequency. So the edges kind of followed in the direction of towards lower frequency always, and fewer subsamples in the...
I think you can have a single cycle. In the extreme case, you can have a single cycle.
Assuming that you have the equal... So, how? So yeah, so that's why the main point here is that we group each node. So if we do have mutations that occur in the same subset of samples with the same frequency, we group them. That's kind of, yes, exactly, to avoid that. So we're guaranteed... And I will, of course, we don't have that every single mutation that co-occurs actually has exactly the same frequency in the data due to the noise. So I'll talk about this in a second. So, okay, so it follows directly from the construction that the true lineage tree, which we assume to satisfy all of the constraints, will be embedded in this graph, right? So some of the edges will actually correspond to the true lineage tree we're trying to infer. Now, so how do we find it? Well, what we do is we find all the spanning trees in this constraint network that also satisfy the third constraint derived from the perfect phylogeny model, which is this third constraint is that now we don't look just at pairwise, just pairs of nodes, but we actually enforce that the sum of the children, let's say here the frequencies in each sample don't exceed the parent. So we generate all the spanning trees that satisfy the third constraint. One of those trees, assuming that the perfect phylogeny holds, will be our true lineage tree. So this is the main formulation. And now I'll go very quickly to basically the main steps of the method. So the first step is, well, how do we group, how do we construct the nodes? How do we group SNVs by presence pattern and frequency such that we assume that actually SNVs are grouped together. They indeed have the same fraction of cells that contain them in the sample. And then the second step will be to construct the network given our groups of SNVs. Final step is to find all the lineage trees in this network that satisfy the third constraint. So very quickly, how to group and cluster. So as I mentioned, we have a naive kind of heuristic approach to determine whether a mutation is in the sample or not, or whether what we're looking at is actually noise. So I'm not going to go into detail because we don't have time, but we basically have two hard thresholds below which and above which we assume that the absence and the presence are robust, okay? And then we formulate a min set cover problem to determine what happens to the mutations in the gray zone, where robust mutations basically are able to pull the guys into their presence profiles that they have. I don't know if that make any sense. I can discuss this further. But as I mentioned also, there are a lot of existing tools that are specifically dedicated for this problem, and we allow users to plug in whatever results they want to the method. So in particular, we allow for each mutation in the matrix for the user to provide an additional column that tells us exactly, is this mutation believed to be present or absent in the sample. So in fact, so I didn't mention this actually. So what we really want for each mutation is a type of a binary profile where we denote with a 1 that mutation is there and with a 0 that mutation is not there. So here we have a germline variant, which is has a high frequency in every single sample. So it has this 1, 1, 1, 1 profile. In this case, we have a variant that is present in sample 1, 2, and 3. So it gets this binary profile. Okay. So this comes to the first way in which we group the mutations, which is by the binary profile. So we collect all the mutations that have the same profile into what we call a single SNV group. So in this case, this mutation will fall into a group of mutations that have presence only in sample 1, 2, and 3. So this guy, respectively, corresponds to these 3 entries. This is, again, on frequency, and so on. So we end up with these 4 groups of mutations. We are focusing on somatic mutations only, so ignore the germline. Now, given these groups, the other thing that we wanted is that the mutations that are present in each node also have similar frequency. We believe them to have the same proportion of cells that they're present in. So we have a clustering step based on VAFs for each SNV group that we found. So first breaking down by binary pattern, then cluster. We use here out-of-the-box EM clustering for GMM. But again, this is another step that we provide a hook in the method for people to use their own clustering, favorite clustering results, and we can operate with that. So essentially, we have our nodes. And now the next step is to construct the DAG that I was mentioning. In particular, given our clusters, each one of them will be a node in our network. And then we will put edges amongst the nodes that satisfy the constraints 1 and 2 that I mentioned. In particular, that there will be an edge if the binary profile of the 2 nodes of the child node is a subset of the parent profile, and the variant allele frequency in each sample is lower than the variant allele frequency of the parent. So let's say here all of the constraints hold. However, here they don't. I believe it's a variant, VAF constraint violation, even though the profile was fine. Yeah. What? No. Oh, sorry. Okay, almost there. So finally, given the network, we search for all spanning trees. This is one tree that we find from this network, and we have actually, I'm also putting here the breakdown of the samples that the tree gives us. In particular, if we were to follow each distinct path in the tree containing only the nodes present with mutations present in the sample, we can get each subclone to be represented by a distinct path of the tree. Here we have this path and then this path, okay, for sample 1. Okay, so very quickly the results. We used a very nice data set provided by Gerlinger et al., CCRCC, data set in which they collected multiple samples from 8 different patients for a total of highly validated 587 SNVs, so this is deep coverage. In their analysis, they used a multi-step pipeline with calling SNVs, clustering, and then running traditional maximum parsimony approach. So very thorough with analysis with some manual work. Anyway, we ran LICHE on the same data set. So these are LICHE results, and these are the trees from the paper in Gerlinger. So for 5 of the patients, our trees were identical. For the other 2, they are extremely similar. And for one case, there's an interesting result that LICHE was able to find. It found actually 1 more sample to be heterogeneous that Gerlinger et al. Did not report. So here is actually a true constraint network, which is very similar to the graph I showed. I modeled it for one of the patients, EV007, for which we got an identical tree. So this is a network. The spanning tree is embedded with the darker edges. This is it. So our results actually flipped from theirs, but if you were to follow each branch, you will find the same decomposition of the samples and the same branching as their constructed tree.
And what did they do to construct their tree? What is the difference?
So basically, they call the variance. So they have kind of a multi-step long pipeline where first they call the variance in each sample, then they cluster samples. This gives them the binary profile. So if you can see here, for sample R3, for instance, they obtained 2 clusters, the dominant and the minor cluster, just based on allele frequency. But now they supplied this to create the binary profiles of each effective leaf. So some of the mutations that are in dominant won't be in the minor. And now using just the binary profiles, they run maximum parsimony. So traditional phylogeny-based approach. So in the next example, I actually have one reason why imposing VAF constraints that we do during phylogeny reconstruction makes a difference. So here we found an additional sample to be heterogeneous, and that happened because actually the group of mutations that were private to the sample occurred with the 0.17 frequency. However, in their tree, the parent of this group of mutations actually was a group of mutations shared between R2 and R4, but their frequency was just so low, at 0.06. So we actually placed this private group of private mutations in R4 as a descendant of a higher node, because the VAF constraint was violated actually here. So this is one example. Very quickly, we also have results in the paper, I won't go into it, from the Bashashati et al. Study that looked at ovarian cancer and also had collected multiple samples. Here's briefly results. And this is, I don't know if I have time for this. No, okay, so while this is running, we have a GUI to interact with our method. It was getting Oscar buzz. No, I'm kidding. I would like to acknowledge everyone on the project, in particular Seraphim and the Batsoglu lab, also a lot of our collaborators in the School of Medicine. This is very 90s looking. We've updated that since then. Anyway, School of Medicine, in particular Rob West, Aaron Sidow. So, okay. And all the collaborators on this project are listed here. Okay. Any questions?
So maybe we should take the questions offline, because I have a gazillion questions, starting with the very basic one that I don't see the difference between this and sit-up. But I think you guys are going to do that. Okay. So we thank the speaker again. You should take the questions offline.